Cell wall active agents inhibit the bacterial cell wall synthesis. They are divided in 4 groups: cycloserine, bacitracin, vancomycin, and beta-lactam. Beta-lactam: penicillin, cephalosporin, carbapenem, astronum. Most bacteria are divided in gram-negative or gram-positive category. For example, Staphylococcus is gram-positive while E. coli is gram-negative. Both bacteria has different cell walls, and that's why the drug effects on one category will not affect on the second category. In gram-positive, the cell wall has a sugar backbone. The sugar molecule is cross-linked with peptide chain. Sugar is also called glycan, so gram-positive wall is called peptidoglycan. There is only one sugar molecule in the cell wall. In gram-negative bacteria, in addition to peptidoglycan membrane, there is another outer lipid cell wall. In gram-positive, just outside the cytoplasmic membrane, there are penicillin-binding proteins. So to attack on gram-positive bacteria, beta-lactam ring has to pass through the peptidoglycan cell wall and attach to penicillin-binding proteins. Peptidoglycan network in gram-positive bacteria is highly porous, so it's easy to reach to penicillin-binding proteins. But in gram-negative bacteria, there is a lipid membrane present outside, which is not highly porous, so big molecules or highly charged antibiotics cannot pass through. Lipid cell wall has protein pores called porins, so any drug to reach to penicillin binding protein, it has to be small enough to pass these openings. Penicillin is divided into narrow spectrum and broad spectrum categories. In narrow spectrum, there are natural penicillin and there are very narrow spectrum penicillin. It means natural penicillins are moderately narrow. Penicillin G given intravenously and penicillin V can be given orally are natural penicillins. Both of these penicillins are moderately large sized. These are very safe drugs. In penicillin, the R ring is what goes unattached to the penicillin binding proteins. If bacteria destroy this ring, they will make the drug ineffective. That's why they are called beta-lactamases. Natural penicillins are effective on gram-positive cocci and bacilli and gram-negative cocci, but not effective on gram-negative bacilli. They have very narrow porins. Natural penicillins do not work on spirochetes. Treponema pallidum, which causes syphilis, is present in the gums, but it's not pathogenic. Very narrow spectrum penicillins are bulky and cannot penetrate into the gram-positive bacilli. They are called anti-staphylococcal penicillin. They have large R group. For example, methicillin, nafcillin, cloxacillin, decloxacillin, flucloxacillin. Staphylococci started to make penicillin cutters called beta-lactamases to destroy beta-lactam ring, and these very narrow spectrum penicillin can stop these bacteria. To deal with beta-lactamases, scientists made modifications and protected R rings, which cannot be destroyed by beta-lactamases. But now the molecule is big and cannot work on many bacteria, but very effective on staphylococcal bacteria. These penicillins are called very narrow spectrum penicillins. Penicillin V should be given to children suffer with rheumatic fever. Now bacteria gets smarter and change its penicillin binding protein called methicillin resistant bacteria. Ampicillin and amoxicillin are wide-spectrum penicillins. The molecule is small enough to pass porin and attach to the penicillin-attached protein. These medications are also good for enterococci, which is a gram-negative bacteria. Helps. Haemophilus influenza, E. coli, Listeria monocytogenes, which is a gram-positive, Proteus, Salmonella can be destroyed with broad-spectrum penicillin. Lipopolysaccharide, which is cytotoxin, is present in gram-negative bacteria. They are part of the outer lipid membrane. Only Listeria cytogenase, which is a gram-positive bacteria, has some lipopolysaccharides. Because broad spectrum are small molecules, they have small side chains. They cannot protect beta-lactam ring base. We can use clavulanic acid drugs like sulbactam, tazobactam, and clavulanic acid with ampicillin and amoxicillin to engage beta-lactamases and let the penicillin work. Amoxicillin with clavulanic acid is called augmentin. Ampicillin with sulbactam is called unesin. Green pus is produced by Pseudomonas aeruginosa, which has tight and few porins or virtually no porins. Anti-pseudomonal penicillins are effective on all bacteria, including Pseudomonas. But their side chains are very small. Some of them are piperacillin, ticarcillin, azlocillin, nazlocillin, meslocillin, carbenicillin. Ticarcillin comes with clavulanic acid called timentin. Piperacillin comes with tazobactam called zoosin. Bacteria's outer layer is made of phospholipids. Two monosaccharide units attach to other two monosaccharide units. First unit is smaller and the other unit is larger. A smaller unit is called NAM and the larger unit is called NAG. NAM stands for N-acetyl-murmic acid, while NAG stands for N-acetyl-glucosamine. There is a short peptide chain attached to the NAM. These repeating NAM and NAG unit repeat again and again and make glycan backbone. Glycosylation reaction attaches NAM and NAG to other NAM and NAG. Outer layer is made of two layers of these sequences where these peptide chains are in the middle and cross-linked each other. For bacteria, its membrane is important because of osmotic pressure it has to maintain inside. NAM and NAG unit combine with UDP, uridine diphosphate, and get activated. This is called murine synthesis. On NAM, different enzymes come and add amino acids and make a peptide chain, and then NAM and NAG will be taken to the cell membrane. In the membrane, there is a lipid-soluble molecule BPP, bactoprenol phosphate, which will remove the UDP, put NAG and NAM together, and flip to transport them out. Glycosylation is when one NAM-NAG molecule is attached with the other NAM-NAG molecule, and it's done by the enzyme on the cell membrane. Transpeptidase reaction is when the peptide chain of both layers cross each other by enzyme on the membrane. Drug cycloserine does not allow the amino acid to build up during murine synthesis and destroy bacteria. Drug bacitracin will stop the flip of BPP and the molecule will be unable to flip NAM-NAG molecule out and the membrane will become weaker and bacteria will die. Drug vancomycin stops glycosylation and NAM-NAG unit stop binding together and bacteria die. Beta-lactam penicillin come and bind with the enzyme responsible for transpeptidation. Peptide cross-linkage stop and bacteria die. Autolysins are protein in bacterial cell membrane responsible for remodeling of bacterial cell wall. Autolysin inhibitors are also present in the cell membrane to stop autolysin to overwork. Penicillin will bind to the inhibitor and let autolysin keep working and they will overproduce cells and bacteria will die. Beta-lactamases are proteins on the bacteria. They have three kinds: Penicillinases, Sphaelosporinases, extended spectrum. Penicillinases break down beta-lactam ring of penicillin. Sphaelosporinases break down sphaelosporins. Extended spectrum can break down both penicillin and sphaelosporin. Beta-lactamases in gram-positive bacteria can be secreted around the bacteria, and penicillin can be destroyed far away from the bacteria itself. In gram-negative bacteria, because of the cell wall, beta-lactamases cannot go out and keep accumulating between membrane and cell wall. This area is called periplasmic space, and that's why gram-negative bacteria are very strong. If bacteria is proliferating, making new cell wall, we can destroy them with penicillin and sphaelosporin. But if the bacteria is not making wall, then it's difficult to get destroyed. Bactericidal drugs which kills the bacteria are penicillin, cephalosporin, vancomycin, aminoglycosides, fluoroquinolone, metronidazole, because they kill bacteria with the help of immune system. Bacteriostatic drugs stop the bacteria to proliferate, but elimination of the bacteria is done